And I also put this slide up to talk about a point once more that I talked about in the lab, and that is the square of the correlation coefficient. The square of the correlation coefficient tells you what? Great, or by the predictor variable. Right. Excellent. So that tells you the proportion of variance that's accounted for, the square of the correlation coefficient. That's one of the things I definitely want you to take home from this little exercise. Okay, all right, so Sir Francis is going to tell us the variance left over after the variance of the other variables has been removed is the residual variance. It's precious to us because it has the variance that's specific to the olfactory bulbs. By the way, we also talked about the fact that yes, you can statistically control for things as we had to do in our study, but is it all, why don't you always do that? If it's that easy, just, crunch some numbers. Why don't you always do that? Why do you bother to construct really careful control groups? Let's go back and look. Okay, so back here at the old conceptual equation, there is an assumption, and the assumption is that each of these components is independent, right? If that's not true, is this going to work real well? No. Okay, so that's one reason why you want to go ahead and have, if you can, careful control groups. We let a lot of things vary in this study, or the folks that made the mouse brain library for us let a lot of things vary because they wanted to look at lots of different questions. So it's not a cure-all, but a lot of times in the clinical setting, for example, you have people come at you in all kinds of shapes and sizes and genders and whatevers, and you can't control for that, and you might want to factor that out. You can do something like multiple regression to control for that. All right. Okay, so we used multiple regression to statistically control for those variables. We also talked about mistakes that can be made with statistics. They come in two big flavors. We talked about type 2 errors, which are false negatives, and we talked about how to go about getting these false negatives. Of course, you don't want that, but how is it that you can end up with false negatives? This is when you should say there's a relationship or should say there's an effect, and you don't find one, okay? So if you have a small effect size, you'll make one of these type 2 or beta errors. So in our instance, if we have genes that account for a small proportion of the variance, we may miss them, okay, because their effect size just isn't big enough for us to detect. Another problem, small n. If our sample of recombinant inbred strains wasn't big enough, we can miss some, all right? And the thing that for us was probably the biggest problem is the variance in scores. The more variance you have in scores, the harder it is to get a significant relationship, and that also means especially error variance. If your error variance is high, then you're going to be making more type 2 errors. So let's just pause for a moment and think about that. So we were trying to replicate a study, Williams et al., which is in your reader if you haven't found it yet. And the way that they quantified olfactory bulbs is they had a technician with a razor blade pop it off and pop it on a scale. One technician, one razor blade, okay? Well, how we quantified the olfactory bulbs is we had all of you take different mice, use that NIH image, and take the successive sections, draw it. Each of you being wonderfully unique individuals as you are, you're all doing it a slightly different way, okay? So who do you think is going to have more variation in scores? Us. What's that going to mean for our data relative to theirs? Do you think we will find more or fewer relationships than they found? Fewer. Okay, so let's translate that into QTL peaks, which we'll be talking about shortly. Do you think we're going to get more or fewer QTL peaks than they found? Probably fewer, okay? Because our error variance, I'm willing to bet, was a lot higher just because of how we did it. Okay, while I'm on the topic of how to make mistake with statistics, there's also another big flavor called type 1 errors, and these are false positives. This is when you say there's a relationship or an effect and there really wasn't. So we know that this can happen, so we guard against it by trying to make our outcomes have a lot of stringency before we'll say they're significant. So, and as I'm going to talk about shortly, we did that likelihood ratio statistic over and over and over and over and over and over and over again, thousands of times across the whole genome, okay? If we do that, that type 1 error compounds, all right? So what we've got to do is make each comparison very, very stringent. Otherwise, we're going to be making a lot of type 1 errors, get a lot of false positives. So I figured it out, and for each point, way back when there were only a few hundred markers, it was like you had to be 0.00006 for each comparison to get to a genome-wide alpha 05. It's probably a lot less than that now. And we also have the suggested rate, which is for each individual point P008, and that would leave us actually with a fair size type 1 error rate for the whole genome, 0.63. But again, QTL is a bit of a fishing expedition, so I decided we'll just live with that. Okay. So, as I already just said, lots of error variants will give us false negatives. Type 2 errors when we do QTL analysis, we probably had lots of error variants. We also, in our tour of statistics, talked about the likelihood ratio statistic, which I'll detail a little bit later in this lecture, and I've talked about in another lecture. Basically, it's a ratio of the variance of all the points to the variance of each of the points around their own mean added together. So it's a ratio of variances. And we'll come back and talk about what the data looked like when it's high or low at a later point. When we got in and were getting ready to do the QTL analysis, what was going to be significant or suggestive was actually was determined by something that's called a permutation test. And these things are becoming increasingly more popular in biology because now we have computers that are pretty fast and powerful. So what we can do is we can use our own data to generate something like a null hypothesis distribution. Did you guys, when you took statistics, study null hypothesis distributions? So what this did was this took the list of markers across the genome and took your values and then randomly assigned your values to a given marker, ran the likelihood ratio statistic for all those markers, and then logged what was the top one, and did that over and over and over and over and over again 1,000 times. Okay, so now what you can see is what the outcome would have looked like with just random data. All right, so from that you can figure out, okay, what are outcomes that only happen 5% of the time with random data, okay? And so that would be the significant one. And you could also figure out, okay, what's the suggestive level? So what that's going to determine are our cut lines for what is significant and what was suggestive when we did our QTL analysis, okay? So that came out of this distribution here. Not based on, say, the underlying assumption of a normal distribution, things like that that you probably heard about when you took stats. Okay, so, and this is what a QTL analysis looked like. This is not your data. This is the data from another quarter, but you can see, as I was mentioning, so that permutation test determined where this red line for significance would fall, and this suggestive line would fall too. And so what we're going to say is any peaks that get over this line here are suggestive, and if we're lucky enough to get any over that line, that would be significant QTL. Did we get any over there, by the way? No, bad news for us. But it's still a happy day. All right. Briefly, in our tour of statistics, and something we will come back to in this course, is an analysis of variance. When we do Dr. Knowlton's unit at the end, we'll also be doing an analysis of variance. Analysis of variance is good for finding the differences among means for several groups. And one of the things that we were interested in is once we cleaned up our data, were there differences among the recombinant inbred strains and the F0s? If there weren't, game over.